She works from home. He's also.
During the folder?
Yeah, right on the desktop, it's 3:30 session,
Okay,
so the folder should be.
Oh, great.
Okay.
All right.
okay, thank you.
You're welcome.
So, yeah, we have, I mean, relative to like 260 hours, that sounds like.
The first floor was fine, is there? What's it going to be?
Yeah, we have kind of like everything. Yeah, this, rural town, so, relatively small wastewater. Right next to the subsewage and wastewater. project. So, and also the ground is impacts on.
We make sensors. Yeah, it's my advisors. There's my PhD.
And that's all. And Peter. And but some of the also.
Really just really so,
But it
really just so
You know, everyone's like,
yeah.
it also used to be called Sweet Sense. And a lot of people. I didn't reach out to anything.
Whoever the CDC was. So now our lab is trying to look at everything all at once, bacteria, protozoa, viruses, so, yeah. That would be helpful, though. We're actually money. But not all of it.
Are you doing any of that like right from hospitals or?
She's very.
No, but part of the stuff, I'm working with clinicians who's doing mailing at hospital. And so at I'm waiting for them. Yeah. And I'll have my
Just say, that's right of the day.
yeah. So whatever level.
Yeah.
Still have 4 minutes.
Like,
Yeah.
chill for
I
like,
see them, but I don't want them to like take my phone.
every time. Okay, guys, we can take. I mean, it's like.
You have a very solid yeah. Yeah, it seems like everyone's trying to figure out how to like optimize methods for how to do everything all at once right now. Oh, I don't know. Yeah. Yeah. Not an option. Right, yeah. You know, there is a lot of yeah.
It's hard, right? Yeah. Yeah.
A lot.
Like, scream. Right. Yeah.
Yeah.
I think everybody.
Yeah.
So hello everyone, good afternoon. Let's start this section. Just for everyone to know where you are, because sometimes I get lost here and I go to the wrong room. So we're in the windflower and our session is about microbial detections. So to double check today, I went twice in the wrong place. So I always do that. So our first presentation will be by Sean Daly. And he's going to talk about microbial source tracking and enteric pathogen detection in humans and domestic animals in urban Kenya. So, please.
All right. Good afternoon, everyone. Again, my name is Sean Daly. I'm a PhD student at North Carolina State University, just down the road from here. I work for Dr. Angela Harris. Today, I'll be talking about microbial source tracking and enteric pathogen detection evaluation that we did in urban Kenya. So what's the problem? Domestic animal ownership is a very common practice in low and middle income countries. They're a very valuable economic resource. And animal husbandry, despite rapid urbanization, is still a very common practice. And this can involve a variety of different animals, thinking cattle, poultry, even dogs can be considered in this classification. And this is important because animals can be kept in and around the living space, outside the living space, in a very close proximity to humans. And the domestic animal ownership has already been associated with pathogen presence in the environment, as well as some negative health outcomes have been associated with ownership of domestic animals in these settings. So how do we evaluate the presence of pathogens or microbial quality in the environment? Very common methods involve the use of indicator organisms, things like total coliforms, fecal coliforms, and on the next level would be E. coli. Another popular methods that are on the rise over the last few decades are molecular methods, thinking primarily PCR amplification. This is when you have a gene target, and through PCR amplification, you can exponentially amplify the presence of this target in your sample to the point where you can detect it. There are some challenges with PCR methods versus indicator E. coli methods. Typically, you can only use one target at a time. It is a little more cost and labor intensive compared to E. coli and other fecal indicator methods. However, it does have its strengths, and I'll be talking about a couple different molecular methods that we used in comparison with E. coli in this setting. So where are we? In the summer of 2019, our team and our collaborators collected with domestic animal, human feces samples from urban Kenya, as well as source and stored drinking water samples. So as you can see here, we've collected fecal samples from chickens, cows, dogs, ducks, goats, and humans. And it's about 20 to 25 samples per host that you see up there. And then we collected about 90 water samples, which include both stored and source drinking water, as well as some blanks collected in the field in 2019. Our first molecular method that we're going to be talking about is microbial source tracking assays. These are host-specific genes that are found in the feces of certain hosts. So we use 3 different targets, avian GFD, which targets avian feces, such as chickens and ducks, the HF183 assay, which targets human feces, and the room two-back assay, which targets ruminant feces, such as cows and goats. So you can see where this overlaps with the feces that we detected. So first, I'll talk about the MST results. This is a little busy. I'll walk you through this figure. On the y-axis, we have copies per DNA target that we detected, normalized by the nanograms of DNA that we extracted. Each column represents the 3 different microbial source tracking assays that we used. And then the x-axis within each column that repeated is the different hosts that we investigated. The points in red are like the target hosts for each assay. So for instance, in the middle column, we have the human feces are one that are targeted in red because, excuse me, that are painted in red because the HF183 assay is targeting human feces. Big takeaways from this figure here. The room 2 back assay had very high detection rates in its target hosts. We have cows and goats. All of our cow feces. We detected the room 2 back assay in all of their feces, and in very high rates in goats, and in very low rates at non-quantifiable rates in non-target hosts. The HF183 is kind of the opposite side of that spectrum. We had relatively high detection in human feces, but it's very similar rates in dog feces, as well as we found the HF183 target in every other type of host that we investigated, except for cows. The avian GFP assay, which targets avian feces, probably a little bit of a middle ground between room 2 back and HF in the setting. We had relatively high detection in chicken and ducks, which are the target hosts, but it was significantly higher concentrations compared to the detection in the non-target hosts of dogs and goats. What does this mean? Well, it's important to validate these assays in certain settings. That means evaluating the sensitivity and specificity toward the target hosts. So as you can see here, again, I've highlighted in yellow what the target hosts for each assays are. Sensitivity and specificity, essentially the rate of detection and the rate of false detection in the non-target hosts. For avian and room 2 back, we had assays in the ranges, or excuse me, sensitivity and specificity metrics in the ranges of 80 to 90%. For HF183, we had it in the ranges of about 70 to 80%. However, like you saw in that figure before, the HF183 assay was detected very commonly in dog feces. So if we can combine this kind of target group into maybe like man plus best friend, human and dog feces, the specificity actually raises up to over 90%. The second molecular method we're going to be talking about is TaqMan array cards. So these are a PCR platform that allows you to detect simultaneously up to 48 different targets. So we purchased the off-the-shelf gastrointestinal card from Thermo Fisher Scientific. And as you can see here, we have 48 different targets, a range of bacterial, viral, and protozoan pathogens, and a number of controls that you can see that I've kind of highlighted. Using TaqMan array cards, we did a pre-amplification step. Since we'll be applying these to feces as well as drinking water, we wanted to make sure that we can kind of raise our sensitivity up to the maximum possible. And following the pre-amplification, we ran the TAC. What did we find? So in this leftmost column, we kind of have the rank order of the average number of pathogens. Dogs, by far and away, were significantly the most pathogens on average per sample. In the far right column, we have the most common pathogen that was detected in each host. Mostly different strains of E. coli. Aside from E. coli 157H7 and enteroinvasive E. coli, the most common pathogen detected in cows was Entamoeba histolica, which is kind of a protozoan pathogen. So what does this mean? This is a very busy figure. This shows all of the results from our TaqMan array card analysis. On the y-axis, essentially each row is a pathogen target. Each column is a host species, so you can see chicken as kind of the leftmost column. And then the darker shaded, the cells where the columns and rows meet indicates the larger proportion of samples for that host where we detected each individual pathogen target. I'm not expecting you to go through each one of these cells right now. I will talk about some specific important takeaways we want to highlight. Pathogen host relationships, there's a significant amount of uncertainty surrounding that area. It can vary across geographies. And if you were to say find adenovirus in an environmental sample, there wouldn't be a very solid grounding in literature to say, oh, this is probably from X host, X animal host. Some relatively accepted host pathogen relationships in the literature, things like adenovirus and norovirus have been identified as human enteric viruses. However, we actually detected them in higher rates in dogs than we did in humans. Things like Campylobacter are very commonly found in cow feces. However, we did not detect any of our strains of Campylobacter in our cow feces. Shigella has been identified pretty recently as a human and closely related primate pathogen as their animal host reservoirs. However, moving forward, as you can see, Shigella was actually correlated with dog feces compared to the other hosts. So not going through all this, you can see dog feces obviously had very high rates of of pathogens per sample compared to the other hosts. So it seems that it would be reasonable to assume that they would have the large number of pathogens that were correlated with them. Astrovirus and Shigella are ones that we could have big takeaways in this space because they're typically not meant to be associated heavily like this with dog feces. So we looked at 3 different ways to evaluate the microbial quality of drinking water. We have the traditional indicator E. coli methods. We just have our microbial source tracking qPCR assays, and then we have the TACMan array cards, which evaluate directly pathogen nucleic acid. In the E. coli, about a quarter of our source water samples contain indicator E. coli, and about 50% of our stored water samples contain indicator E. coli, and they range pretty significantly in their concentrations. Microbial source tracking markers, we had pretty low detection across the board. The HF183 marker was detected in one source and one stored water sample, but they actually weren't connected to each other. So the source water that was contaminated, or excuse me, the source water that we detected HF183 in did not lead to the stored water sample that we detected that marker in as well. So for the tax, between about 20 to 30% of the water samples, depending on source were stored, had at least one pathogen target that was detected. Source waters and stored waters both contained Aeromonas hydrophila, and then we kind of have a different range of pathogens that were detected in source waters. What I want to highlight here moving forward is that we did detect some pathogen targets in samples that we did not detect indicator E. coli in. Unfortunately, our sample sizes were relatively small, so none of these correlations were statistically significant. There's also an issue of enumerating E. coli and those actually growing organisms that are what is the word? Essentially, the molecular methods, they don't differentiate between organisms that are active and organisms that are inactivated. So there is a little bit of a limitation there with applying, saying, oh, the TACMan array cards detected pathogens in samples that did not contain E. coli. That doesn't necessarily mean that there was active pathogens that could cause disease in those samples. However, it is interesting that you would see pathogen nucleic acid markers in these samples. So big takeaways. We successfully validated the room 2-bac and avian GFD assays in this setting. The HF183 assay was significantly less sensitive and less specific. However, the specificity was raised up to over 90% when you combine dog feces in that target group. The TACMan array cards offer a feasible method for detecting multiple pathogens in environmental samples, including drinking water, where you would expect these kinds of contamination to be at pretty low concentrations, will be a little bit harder to detect. As well as our results in this setting don't necessarily agree with the established literature on what types of pathogens are strongly correlated with what types of hosts. And we really highlight the uncertainty in that space. And we offer kind of TACMan array cards as a very viable option in order to address that knowledge gap. We compared multiple strategies for evaluating the quality of drinking water, including indicator E. coli and 2 molecular methods. We kind of highlighted the strengths and weaknesses of both, talking about viability versus inactivated cells. We detected pathogens in samples that did not contain E. coli. And we kind of offer TAC as a molecular method that could overcome limitations seen in FIV and MST assays, things that like indicator E. coli are not necessarily the disease-causing agents, where TACMan array cards are capable of detecting specific pathogen targets, even in low concentrations in samples like drinking water. Thanks so much. I'll be happy to take any questions. Any questions? Yes.
Mark Thompson, University of North Carolina. Very interesting. So one of the things you had on the last slide at the university was that the molecular methods provide to make determinations of risk. I'm concerned about that statement because it's a molecular method that only detects genes and doesn't detect infectious organisms. So I would recommend qualifying that statement because there's no consistent relationship between the concentrations of culpable or infectious pathogens and gene copies. The relationships can vary by several orders of magnitude.
Yeah, that's certainly a limitation that's just inherent in the molecular methods like this.
You had an opportunity, and you still do, to look at some of the pathogens that can be detected, because they're relatively simple, straightforward. One of which would have been Aeromonas hydrophila, which is readily culturable and could be detected not only by microdetectors, which is in the party, but it could have been rarely cultured because there's specific media. And there may be some specific media, even for some of the E. coli strains, like the hemorrhagic, because they're media that have been designed specifically for that. So I think in the future, it would be worthwhile for that to be looked at.
I agree.
A better sense of what the relationships are between the molecular signal and the culturability of those represented. Yeah, obviously trying to do this for school level practices.
Yeah.
Or impossible. But I don't know where you were on your PhD. I need someone else in that.
It sounds like a great job. Thank you so much. It sounds like a great next step. That's definitely the trade-off when you come to culture methods versus molecular methods. The culture methods, some organisms can't be cultured. Molecular methods, it's easier to detect things like enteric viruses or RNA targets that are a little bit more tricky to culture, but then you do have the viability versus inactivated cells issue. It would be really interesting to look at specific pathogens in that setting too.
Just one more point. So you found a lot of things in dogs. First,
Yes.
dogs live closely to people. Some dogs, frankly, just like to eat people.
Yes,
And
yeah,
eat food.
that is actually, there's,
That could be the source of why they picked up things like noroviruses.
yeah, and that's a great point. If you're trying to dive into the literature and look at dog feces as a challenge for these type of molecular methods, it's not widely investigated, but kind of the one point that tends to come up is that dogs consume the feces of other animals, that could be exposed to those genetic markers that way.
Thanks again.
We're at time.
Can I ask a question?
Sorry?
Can I ask a question?
Yeah, sure.
A question for a lay person in this case. Why to do this experiment in Kenya? I mean, you are looking for testing those things in animals that you can find everywhere. So why to go all the way to Kenya to do this experiment?
Sure. So we were looking to investigate what kind of exposures to pathogens that these individuals in Kenya may be facing due to the domestic animals that they have in close proximity to. Obviously, we just discussed kind of strengths and limitations of these molecular methods and the indicator methods. But these are the kinds of settings where people will be routinely exposed to animal feces. Someone like me, I'm not routinely exposed to animal feces. So the risk of zoonotic pathogen transmission is very small. Yeah, so I hope that answers your question.
Okay.
These are kind of the areas where these issues are prevalent.
Okay.
Thanks so much for your attention.
So our next presentation is by Emily Bedell. She's going to speak about development and validation of a highly accurate PLF sensor coupled with a machine learning model for the detection of fecal contamination risk. Oh, it's very difficult for one native speaker. I can do that so fast.
Where's it's the presentation?
Put in the presentation mode when you're
Cool. Thanks everyone for coming. My name is Emily Bedell. I am a environmental mechanical engineer now working at Verity. I did this research for my PhD at CU Boulder. I did my PhD in environmental engineering, but my background before that is mechanical and I can't let that go. So I've developed and validated a TLF sensor coupled with a machine learning model for the detection of fecal contamination risk. So I'll go over the design of the sensor, why we made some of the decisions we made, then characterization, figured out its feasibility in terms of varying water quality. And then the field validation that we did with the sensor. So as we all know, and we've all been talking about a lot of this week, microbial enumeration, the current methods have tons of limitations. They're time consuming, they're expensive, and they have high training requirements. All of these things lead to a high possibility of miscontamination events. Most of the time, by the time you figure out that water is contaminated, people are already drinking the water. So because of this, I think about 6 years ago, I don't know if Esther's in here, but UNICEF put out a target product profile for a product that could find E. coli faster in drinking water. So they want a product that can have false negatives and false positives below 10%. They want a limit of detection of 10 CFUs per 100 milliliters of E. coli. And they wanted to distinguish between the WHO risk categories. They want less than 3 hours from measurement to result. They want it to be easy to use, battery powered, lightweight, and portable, and cost between $1 to $5 per test. So this was the initial motivation for the research. So our possible solution to this is a tryptophan-like fluorescence sensor. So fluorescence occurs when a molecule absorbs a photon at a specific wavelength. That wavelength is then bumped up to a higher energy state, and once it falls back down, it emits a photon at another wavelength. So in practice, we do this by shining UV LEDs on water, and then if there is microbial activity present, it will fluoresce, and we pick that up with a light reader, like a photomultiplier or a photodiode. So there have been multiple studies that show the relationship between TLF and fecal contamination. This one on the right was done by Baker and colleagues, and they used a portable fluorimeter to assess the relationship between fluorescence intensity in E. coli and got a correlation of R equal to 0.74. The one in the middle was studying the correlation between TTCs and TLF in groundwater. And the important outcomes from that study were that this method will work better if we use it to output the risk levels of contamination. So instead of saying the output from the sensor means there's 10 CFUs per 100 milliliters, the output from the sensor means there is low risk contamination or high risk contamination, because it's not precise enough to output those exact numbers. And then, yeah, so with the portable fluorimeter, this study was able to get a false negative rate of 15 and a false positive rate of 18%. So what's wrong with the technology that exists right now? There are a couple fluorimeters that people are using, but most of them are probe fluorimeters, which require calibration, constant cleaning, and you still have to use grab samples. There's this one flow through on the market, but I can't really tell if it is for tryptophan-like fluorescence, and it doesn't seem to have any remote reporting or be calibrated for E. coli. So here's where there's technology development opportunity. We know that TLF is correlated with fecal contamination. There are a couple sensors on the market, but they lack some key capabilities. So we wanted to develop a sensor that would be inline and remotely reporting. And then we know that all of these parameters will interfere with the fluorescent signal. So biofilms will grow on the sensor surface. Mineral scaling will impact the signal, pH, interfilter effects, water temperature. Humic-like fluorescence overlap, all of these things will cause false positives and false negatives from the sensor. So we wanted to try and use machine learning and then be able to output a risk level from the sensor. So for the design, I wanted it to be autonomous, in situ functionality. have a high sensitivity to tryptophan concentrations, simple maintenance and cleaning, some range and gain levels that you might be able to choose that could be applicable to either surface water or piped or groundwater. And then we wanted to detect and measure proxies for fouling we could input into the machine learning model. So at this point, we did a bunch of testing in the lab and found out that it would be possible to make a sensor that would give us the sensitivity levels and published that in sustainability. From there, we moved from a sensor in the lab connected to power supplies and oscilloscopes to a field deployable sensor. So this has a little peristaltic pump in it that pulls water up through a quartz cuvette that has the UV LEDs around it and a photo multiplier. This is connected to an amplifier with a LTE connection on it that sends the data. And then there's also a water temperature sensor, and it has a solar panel and battery in there. This is from a test we did in the lab to find out the lowest sensitivity to E. coli we could get. So we made dilutions with wastewater effluent, and we were able to detect 10 CFUs per 100 milliliters of E. coli. There is one drop once you get around the 1,000 CFUs per 100 milliliters, and that's most likely due to increase of turbidity and but we're thinking around those levels. Ideally, your sensor's already alerting you that there is contamination. So then in the lab, we characterized the sensor to changes in pH, which outside of levels from about, I think it was 5 to 9, we didn't see changes in the signal. So in normal drinking water levels of pH, the sensor would be okay. The increase in water temperature does decrease the fluorescence output, so that's why we included the water temperature sensor. And then turbidity has varying impacts depending on the makeup of the particles in the water. And then we also characterized for fouling, which I'll show in a second, and we characterized for scaling. So to see biofouling impacts on the sensor, we put a few sensors into a bucket and then dosed them with wastewater effluent every so often. So if you could tell from this graph, we put wastewater effluent in the bucket, and then you can see the signal slowly start to increase. But when we dose it again, the signal increases drastically again. So this is where the machine learning model comes in. If the signal is increasing at a certain rate, it would be able to figure out that this is not contamination, this is increased due to biofouling. And we do that by inputting time since last clean into the model. So then we did a field validation where we installed 4 sensors on the Boulder Creek last summer for 2 months and developed a training set for the machine learning model. We collected samples from the sensor 3 times a day for those 2 months. So the sensor was taking readings every 10 minutes, and we would go take a sample of water from right where the sensor's input was, take it back to the lab and plate it for E. coli and total coliforms and be able to develop that training set for the machine learning model. Some field challenges: the creek flooded at one point and knocked all our sensors over. Sometimes the inlet into the sensor would get pushed into the dirt and start sucking up dirt as well. So we now know that we definitely need a filter on the inlet of the sensor. This is from the field samples we collected, and there's definitely poop in the creek. We have our highest amount of samples were in the high risk category, but the important thing from this is there's about 50% of samples were above that high risk category and 50% were below. So that helped us when we developed a model to distinguish between not high risk and high risk contamination. The data we put into the machine learning model were the fluorescence output, the hours since cleaning for the biofouling proxy. We put rainfall data in, which ended up not mattering that much because in Boulder summer, it doesn't rain. And then we had 3 relative voltage data, so percentile difference from 7-day average and Z-score. We had sensor temperature, water temperature, and then to try and get some seasonality, we just used day of season, so counting the days since we started. This is the output from the dichotomized high risk machine learning model. So that's either high risk or not high risk contamination. We had an accuracy of 83%, a true positive rate of 80, true negative rate of 86. So that's false positive of 20% and false negative rate of 14, which is above what UNICEF wants at the 10 and 10. But we predict that when we develop this model on either groundwater or piped water, we would get much better accuracy because there is much less background fluorescence noise that will be interfering. This is the variable importance plot where just quickly you can see that fluorescence was the most important variable in the model and the hours since cleaning was the second most important variable. We also did a categorical risk of contamination model where we did output each of the WHO risk levels. So very low, low, intermediate, high, very high. At the very low and low, the model doesn't work because we had such low amount of samples in those categories. We have a patent on the sensor that will be issued, I think, in the next couple of weeks. And then we just published this paper this summer on the development of the machine learning model and the characterization. Some quick reflections I've thought about throughout this research. So if we're thinking about E. coli, its utility as an indicator, if we're developing this machine learning model with E. coli as a training set, we're not really getting away from using E. coli because it's still relying on that measurement. So maybe developing a machine learning model with different methods that might be more accurate in actually showing fecal contamination. We need to do more extensive turbidity testing. We do know that we could add an absorbance measurement to the sensor and it would make it more accurate, but we're trying to cut down on the amount of things that could break or just trying to keep it as simple as possible. And now we're developing a model with piped water. So I've got 4 sensors on the pipe in the lab and we'll be dosing it and building another model. And then thinking about the overall health impacts. So we know that low risk contamination doesn't often lead to diarrhea unless it's in sick or very young or very old people. So where do we need to know if there is very low risk of contamination all the time? Or could this sensor just work as a warning system of high risk contamination because that's what actually impacts our health? Also moving forward with the design of the sensor and productization, because we're trying to get to the market in the next year, we're thinking about feedback informed accountability. So how will the sensor work within communities or for consumers? We need to develop that feedback mechanism. Is it going to be an app? And then thinking about how cost effective, how transparent the data is, who's using it, what's the action when you see an alert on your phone. The end goal of this is to improve the provider or consumer's capacity to detect, manage, and mitigate fecal contamination. So right now I'm working on the next iteration of the sensor. I have it here if anyone wants to see. We're working on entering the market for well water users in the United States. So people that have treatment systems in their own homes could install this sensor and make sure that they are getting clean water, or if they need to change their filter or maintain their treatment system. And we also, a new feature on this is that it will have a cleaning mechanism. So every so often, ideally, the app would tell you you need to clean it. So you squirt a little bit of bleach into the sensor and then tell it that you've cleaned it so that it can put that into the model. These are all the students and people that helped me with this work. Any questions? That's me and Carl in the same place.
Really nice presentation. I'm curious if you've considered using other songs that can collect other water quality parameters real time, and if that would maybe help your machine learning algorithms to take in the fluorescence from the tryptophan sensor and make it a better predictive,
Like putting,
like
Like
pH or,
pH or-
you know, like these other things that you're saying impacts of how
yeah,
it
totally. So
measures, and so they can, maybe correct.
yeah, I think any of those additions would be helpful and make it more accurate, but like I said, we're trying to make it as simple as possible because we know that these things will break eventually. We know that sensors degrade, that pH sensors need calibration, and trying to cut down on the amount of calibration needed, so it can be as simple as possible. That makes sense, but yeah.
Right, nice work. A couple of questions, one in particular. But comment first. E. coli may not be the best indicator of groundwater quality in some places like this in North Carolina. We did a study where we found almost no E. coli in the groundwater wells, but we found a lot of holy flashes. These are the viruses. Your sensor probably didn't be able to detect those. Maybe even not, but it would require a different target.
Yeah,
But another bacterial group that's very important is hidden indicators and pneumococcus, which are actually more persistent in the environment and are relatively eco-specific. So might we not consider developing a sensor for an enzymatic marker for pneumococcus? I think that might be worth exploring at some point. And let's see, we should go listen to this. Maybe with the
yeah, I think looking at the fluorescence marker of these different things, because this is a specific, tryptophan-like fluorescence and indole comes from tryptophan, which is in these enteric pathogens, right? So that it's not just E. coli that will set the sensor off, but yeah.
Okay.
Just curious,
unit cost and the operating cost if you're replacing sensors, et cetera.
Yeah, so the cost we are shooting for is $1,000 per sensor. That's probably as low as it could go. We're trying to get lower, but right now the most expensive component is the quartz cuvette. And I've tried other types of glass, cheaper types of glass, and nothing gives us the sensitivity that we need, but operating costs, ideally, it can just be installed, and then we will have some back end, so the data will be sent to the cloud for the model to work, so there will be something there for sure. Thanks.
I know there are a lot of questions for her, so I think she'll be available later for the fun. If anyone wants to ask more questions to her. The next presentation by Gautami Rao. She's going to talk about beyond COVID-19. simultaneous quantification of many pathogens in wastewater to inform public health response. Stephanie? Presentation mode.
Great, okay. Thank you. Welcome, everyone. My name is Gautami Rao. I'm a PhD student here at UNC Chapel Hill, working with Dr. Joe Brown. And I'm going to discuss, our simultaneous quantification of several pathogens that we detected in some wastewater samples that were collected. So wastewater surveillance, as sort of a general background, first began in the 1940s for poliovirus and has expanded over the years to include Salmonella typhi, which is the cause of typhoid fever, pharmaceutical and substance abuse markers. So there are several terms the field uses, and WBE, or wastewater-based epidemiology, is one of them. Wastewater surveillance, sewage surveillance or sewage monitoring. And these all refer to the tracking of targets in wastewater, primarily focused on human excreta through traditional wastewater systems. And sort of recently, and also through historical kind of evidence, it's also a public health tool that can provide a snapshot of the health status for entire communities. This is regardless of access to healthcare or clinical testing or participation in surveys. So it kind of gives us a really good understanding of what can be targeted when people are presenting or not presenting symptoms. And current wastewater-based epidemiology efforts focus on a variety of categories. There's the exposure area, where researchers are interested in looking at a number of markers, biomarkers, pathogens, antimicrobial resistance, and identifying detections of what could be potential exposures. Also, the sample collection efforts. So what type of wastewater sample is collected, or what aspect of the wastewater component are we focusing on? Processing efforts on concentration. So which concentration method are you using, if any? Since environmental microbiology often requires taking a larger volume of your sample, concentrating it to increase the likelihood of detecting your target. And in addition to the processing, what sort of quantification platforms are being used? Is it a qPCR or real-time PCR effort or digital PCR? And the ongoing efforts on data analysis and data interpretation. So how exactly are we learning from this? Is wastewater truly being an early warning sort of tool that we can use to understand the spread of variants or pathogens, or the increasing and decreasing trends over time? And then our wastewater infrastructure, such as how do sewer systems or sewer connections differ from on-site sanitation? And what are some of those challenges? And the reality is that our field is expanding so quickly in this area that each of these kind of components have some room for improvement. And with that, our team was interested in looking at wastewater influent that was collected from 4 wastewater treatment plants in Atlanta from February through October of 2020, with the primary intention of looking at and detecting and hopefully quantifying SARS-CoV-2 in addition to other pathogens of interest. So we collected about 34 different 1 liter influent samples, and we concentrated these samples through 2 methods. The first was using the InnovaPrep concentrating pipette, as you can see here. And we took approximately 150 milliliters of our 1 liter influent, and we spun that down. And the supernatant was then pulled up through the InnovaPrep and backflushed, and we had used that sort of backflush as the input into our all-prep power viral kit. We also preserved the pellet that was actually with that entire sample and performed 2 additional extraction methods, the Kaya AMP 96 viral kit and DNA Parcel Pro. In addition, with the remaining about 750 milliliters, we performed this a skim milk flocculation method. So this was adapted from the paper by Philo et al. At University of Washington. And we added some additional skim milk solution, had it shaken up for some time, centrifuged that down, and focused primarily on the pellet with that particular sample type. We then proceeded to do our nucleic acid extractions. As I mentioned, the pellets that we had collected, we focused on using the DNeasy PowerSoil Pro and a KaiAmp 96 viral kit with our KaiCube HT, and the supernatant from the Innova prep was using all prep. And finally, we looked at two different workflows on our molecular methods. We ran a portion of our samples through the reverse transcriptase real-time PCR. So as mentioned in the previous presentation, we also use TAC or TACman array cards, which can do up to 48 enteric detections. And we also performed digital PCR on sort of the subset of these samples to look at a more specific and sensitive detection of a few targets that we had of interest. And the digital PCR platform is actually recommended by CDC as sort of the wastewater-based metric or tool to use. And I think, our qPCR results may also be of interest. So first, I'll show this particular result image here. We have on the y-axis our log10 transformed gene copies per wastewater influent. And on the x-axis, we have our gene targets of interest. I think it might be a little small, but we have on the far left, B-cove or beta coronavirus. This is what we used as our process control. So this is what we had initially spiked at the very beginning to see kind of how our recovery was resulting within this particular extraction method. We had mtDNA or human mitochondrial DNA. We were interested in looking at this potentially as a normalization marker that can be used, or sort of a stronger indicator of understanding the sort of the abundance of human associated contamination there. And our N1 target for SARS-CoV-2. So we saw quite a low percentage amount of abundance of this particular target. And PMMOV or pepper mild mottle virus, which as you can see is quite high in this concentration, but is often used as again, as the normalization marker or this indicator of fecal strength. And we were interested in performing the digital PCR on these particular targets to get an understanding of how can we interpret our data based on perhaps using these different normalization markers. And here are our results for our qPCR or RT-qPCR method. On the y-axis, we have our log10 gene copies per liter of wastewater influent. And on our x-axis, we have an array of pathogens, bacterial, fungal, helminth, protozoan, and viral targets that we were able to detect. Now, I should mention that this particular box plot graph is showing the skim milk flocculation method using the KaiAmp 96 viral kit. We found, I would say pretty interesting results. I think I was a little surprised at sort of some of the concentrations that we were seeing, and then also some of the helminth detection. So we have Ancylostoma duodenale here, which is hookworm. We also have human threadworm here as well. And something that I thought was really interesting was that we have norovirus G1, G2 detection. And during this time when these samples were collected, there was no known outbreak for norovirus. So to be able to kind of identify this even through the molecular methods could be an indication of sort of an unrepresented a clinical manifestation, but only able to be seen through the wastewater, not so much through the clinical if it was never reported. But we did not actually detect SARS-CoV-2 in this as well. So we had no detections in our dPCR. We didn't have any detection here. And I thought, okay, perhaps maybe we weren't looking at the right extraction method here. So what we did was we re-extracted those skim milk pellets using the D&EZ Power Soil Pro and found comparable detections across the board between most of the targets. But we also noticed there's a higher concentration that we were detecting here. So this extraction method seemed to be very sensitive to our particular pathogens. And we also found our hookworm target here. And then we also detected Hymenopus nana, which is dwarf tapeworm, Strongyloides stercularis, which is human threadworm, and then also trichoris. So quite interesting that we're actually detecting sort of these perhaps neglected tropical diseases or helminth detections within an Atlanta-based population. And we were able to detect some SARS-CoV-2, not in all samples, but this is also a preliminary box plot of our results. So we were able to detect SARS-CoV-2 within these samples as well. I think that there's kind of a lot to learn here. How we interpret this data and kind of the next steps, I would say, are still things that we're working through. But what I found interesting was of the 38 pathogens that we had sort of like on our card at this point, 31 or 82% we did have detection for. So in terms of our future efforts, TAC or TAC-based approaches for potential wastewater routine monitoring could be a valuable tool that could be implemented within departments of health. Particularly if we're looking at this from developed countries, this may be a valuable asset to be had. But also, I think the long term here is looking at how can wastewater monitoring be sort of a sustainable approach, even from a global perspective. I mean, if this would be effective at being able to identify multiple targets at one time, can that actually be, a feasible approach moving forward? And a lot of the research and current work is also looking at back calculating up to like a population burden of what this might be. And I caution and I hesitate around kind of doing some of that work. I think future steps here are potentially looking at translating these gene copies to organism concentrations and perhaps seeing like what kind of abundance we can have there, but not necessarily taking it all the way up to the population, since there are several other uncertainties that would be associated with that. So with that, I'd like to acknowledge everyone in the Brown Lab, Dr. Erin Bivens at Louisiana State University, and also Chinghua Wang at Georgia Institute of Technology for their help. So, with that, I'm taking it.
Hi, I'm Amanda. I'm a PhD student at Tech. Really great presentation. I was just wondering, you said you use the CP Select for your wastewater. How much volume were you able to get? And did it just take a really long time due to clogging? And I guess the other question I had was, do you think the skim milk flocculation method was picking up the solids in addition to the viruses in the liquids? And could you talk about that more?
Yeah, so I mean, I feel like we all probably go through this, like how do you pick which method to go with and move forward, and it causes all sort of angst. But for the InnovaPrep concentrating pipette, I thought initially I had planned on, I thought it could kind of pick up more volume of the influent, and it could only, like the maximum I could basically get was like 149, mils. So I just started to aliquot that amount aside, and once I centrifuged it down, sometimes it would take like 2 efforts or 3 efforts, but I always tried to limit the amount of processing time to be like less than 20 minutes because I just didn't want to have too much benchtop time. And yeah, it was definitely a challenge. There were clogging issues, tried to reach out to them. I think they now sell like another pre-clogging filter item you can do, but of course it's like one more thing to have to buy. So that's why I ended up resorting to, if we have this, about 750 mils left, what can we do with it? And I was debating using PEG precipitation versus skim milk. And I think I had just gotten some guidance on just considering skim milk population as an option, and I tried it, and it worked out really well. So I recommend it because I've had some good luck with it, but I know folks who are interested in doing more sequencing based work are concerned about, you know, having these sort of skim milk potential byproducts or whatnot to maybe interfere, and are considering more of the PEG precipitation option. I think both actually are probably quite reasonable. I think I answered.
Nice presentation, and I know how much work goes into making those types of figures, so yeah, really nice. So I'm curious with your SARS-CoV-2 target, because you know, with the tech platform, it's really nice that you can look for multiple targets, but you might lose some sensitivity. And also like how you do the standard curves and all, you know, like there's other kind of challenges with really quantifying using the tech platform. And so I was curious if you looked at your results of the SARS-CoV-2 using your different like ddPCR, qPCR, and TAC. And like, did the TAC, do you feel like it performed sufficiently well for kind of using the trends calculations that the CDC is wanting to use with wastewater surveillance? Like, could TAC meet some of those needs, or do you think ddPCR will still be what would need to be done to get the trends?
Yeah, I think that's a great question. One thing all of this work has really made me think of is just how important it is to connect the right sample matrix with your extraction, with your platform, because even running the dPCR on sort of the supernatant of the influent, there were very low, it was very low in detections. And I think that's because what we're identifying, or what I think we're seeing, is that there's a much stronger association with the solids fraction. So I think if we are considering sort of going down the route of like, where are we most likely to detect SARS-CoV-2, we should probably be focusing on the solids fraction. And I think the sensitivity of TAC was, and it was, the CQs were about 34, so it's still a bit later, but I think for environmental samples, like I would still consider that to be, you know, reasonable.
What was it relative to your qPCR though? Was it like comparable level?
So that was the qPCR. So I think the digital PCR was just very low. And again, that there were slightly different matrices. So I only did the dPCR on the supernatant, and then I did the qPCR on just the solids portions. So there's a little bit, I can't fully do the full comparison there. It would be interesting, but yeah.
With TAP, you've got a ton of things that you're looking at the same time. Do you think that's useful? You know, how do you choose kind of what to focus on? And do you think that those targets which are developed really a lot thinking about exposure, something what we want them to mean in the context of monitoring and understanding disease burden? It's a pretty open-ended question, but I'm curious how you're thinking about that.
Yeah, it's definitely on my mind, and I think that's been one of the challenges, is how we appropriately interpret what this information could mean. I think it depended on the population. So for me, when I was looking at this and seeing some of the detections, to see that in sort of a metro Atlanta area was something that I just was more surprised by. So I thought that perhaps there's space here to understand where we could have targeted public health interventions that may be more relevant. But I think that, yes, even though in each TAC well, I think it's like 0.6 microliters that ends up kind of getting processed, is a little scary to think, but it doesn't, there's so much involved in, I think, the actual preparation of getting those cards to function the way that they do, that I think it's more of understanding how we want to interpret it on the other end. So even if it's not exposure and it's more pathogen like detections and concentrations, I think we have to kind of pull that a little bit further and say, okay, how can we sort of triangulate this back now? Is there a way in which we get this information now? Should we consider sort of a more targeted approach and where we're looking at this, and perhaps use that as sort of a backtracking method? So we screen across a lot and then maybe start targeting. So, I mean, I thought, yeah, I don't also know if this may be a part of the burden, if there's, maybe more homeless people that somehow, you know, we've been able to collect that kind of wastewater, because these wastewater treatment plants, I think it's purely human waste. There's no combined, overflow or any other information, or, well, it depends on which one. And I think the ones that we looked at, I don't believe that was the case.
I'll say that I think, just to quickly finish up, I think for the public health use of the data, being able to dig into for each of those, you know, what is it really gonna tell us about population burden is important then for the uptake of the data.
Mhm. I'd say.
Last question, or you cut me off?
I don't know. I don't know.
Okay,
Use some favorite examples. You have some that are like, now that you've done this, could you consider doing a select sample where you do a direct extraction, where you do these certain things? And I think you're gonna lose stuff at each of the processing steps, and there becomes a trade-off of how much effort versus your return, and concentrate down, but you've got concentrate inhibitors lost during whatever step. There might be this now on those targets of interest with the particular samples, you have some really cool data sets you could put out.
Yeah, I did look at actually a direct extraction method as well, and was not finding, you know, like I kind of did a small portion, but it made sense because the volume was just, you know, a mil or so, which is a lot smaller than concentrating down, you know,
Yeah, you can also
150.
spike it and see what your loss is, but then we can talk about that later.
Yeah.
That's very cool.
Yeah,
Thank you.
thank you.
okay. Thank you, Anthony. So let's enjoy the last happy hour of our conference. More people are having on, so enjoy.
Yeah.
The other virus, yeah, or it was the only virus that.
There is, there is detection or
That's more colleges anyway. Type of virus. For those.